Hello everybody, so today I will be talking about membrane structure and specifically the role of membrane lipids in forming the phospholipid bilayer of cells. So membrane lipids contain a hydrophilic head and a hydrophobic tail. So they're represented like this. A circle represents the hydrophilic head, and you have two lines which represent the hydrophobic fatty acid chain of the tails. So the phospholipid head group contains a choline, a phosphate, and a glycerol, and this is hydrophilic because it can hydrogen bond with water. The hydrophobic tails contain hydrocarbons, and this cannot bond with water, therefore it is hydrophobic. Fatty acids can either be saturated or unsaturated. So saturated fatty acids contain two long tails with no kink in the middle. However, unsaturated fatty acids contain a double bond. Therefore, there is a kink in the middle of the chain. The result of this kink is basically that in membranes that have more unsaturated hydrocarbon chains, these membranes are more fluid because, as you can see, there are big gaps between the lipid molecules. However, if there are mostly saturated hydrocarbon chains in the membrane, then the membrane is more rigid. Also, you can see that because these chains are shorter because of the kink, the membrane is thinner. And when you look at the saturated hydrocarbon chains, you can see membranes containing mostly saturated hydrocarbon chains are much thicker. Membrane lipids are amphipathic, which means that one part of it is hydrophilic, namely the head, and the other part is hydrophobic, which is the tail. And this is very important because hydrophobic molecules tend to cluster together like this, and water basically forms cages around these hydrophobic molecules. So say you have a hydrophilic head group and a hydrophobic tail. This becomes surrounded by water cages. So in order to exclude the water, the lipid molecules will come together to form clusters to try and exclude as much of the water as possible. So if these lipid molecules have a single tail, they form a micelle, whereas if they have double tails, they will form a bilayer structure. So how does this work? So in these micelles, what happens is that in order to exclude water, these single-tailed lipid molecules, what they do is they pack together to form a sphere. So you can see here it's kind of triangular, so they kind of fit together like cones. What happens in the lipid bilayer, where the chains are double chains, so you have two chains here, is that the area is kind of cylindrical, so they pack together to form a lipid bilayer and not a sphere like this. However, you may have noticed that this part of the lipid bilayer is obviously hydrophobic because it contains the chains. However, this part is still exposed to water. So how do you overcome that? Well, this is then energetically unfavorable because lipid bilayers, or the fatty acid tails of lipid bilayers, don't like to interact with water. So what happens is they fold up to form a sealed compartment, and this is energetically favorable. So does this look familiar? Yes, this is the plasma membrane that surrounds the cell. Remember, the cell is three-dimensional. So these are all the phosphate head groups that you can see. Cholesterol is also sometimes present in the plasma membrane, and that also has a polar head group and a nonpolar hydrocarbon tail. Polar head group, nonpolar hydrocarbon tail. And what it does is it basically fills in the gaps between the unsaturated lipids. So you can see where the membrane should have been more fluid because of the packing of the unsaturated lipid molecules. What happens is that the cholesterol sort of sneaks in between them, making the membrane more rigid. So if there's more cholesterol in the plasma membrane, that membrane will be more rigid. Now, do these membrane lipids move? Yes, they do, and you can study the path of lateral diffusion by labeling these lipid molecules. And this is basically how they move. So they start here, they go around here, and then they finish here. However, there are barriers to free lateral diffusion of these molecules, and these barriers are called lipid rafts. So basically, lipid rafts are tightly packed microdomains within the plasma membrane. They're enriched with cholesterol and lipids, and the cholesterol actually holds the raft together. And as you see, membranes which contain lipid rafts are much more thicker, and also they are much more rigid. You can see lipid rafts using atomic force microscopy. So you see these little peaks here? These peaks are the lipid rafts. So basically, what an atomic force microscope can do is that there's this little cantilever that moves over the surface. And when it encounters a lipid raft, it sort of produces a little bump because it moves up. And this is detected by a laser. So here you see the cantilever moves up, moves down, and detects the lipid rafts here. So this is basically the picture that you'll see of the lipid rafts. Another important component of the membrane is obviously membrane proteins. And membrane proteins can be isolated using the freeze fracture technique, where you basically freeze the membrane, pull it apart, and you can see the proteins embedded within the plasma membrane. So membrane proteins can be integral membrane proteins, which can be removed only using detergent. They cannot be disrupted by a salt wash. Why? Because they're integrated within the membrane. So here you can see an alpha helix, a transmembrane alpha helix. So these proteins usually have transmembrane alpha helices, or they can have beta barrels. There are also lipid anchored proteins, or there's basically an alpha helix which is anchored within the lipid bilayer. Now, peripheral peripheral proteins can be disrupted by a salt wash because they're not embedded within the plasma membrane. So here you can see a lipid anchored protein. There are non-covalently attached proteins which are attached solely through ionic interactions. Therefore, you don't need a detergent to disrupt the lipid bilayer to remove these proteins. They can usually be disrupted by a salt wash. Membrane proteins can also move. They can laterally diffuse, and they can also rotate, and this is very important for the function of transporters and receptors as well. However, not all proteins are highly mobile. there are certain cytoskeletal proteins, such as spectrin, which, well, they don't need to move because they provide the scaffolding or the structural integrity of the cell. So, in summary, we can say that the lipid bilayer provides the basic structure of biological membranes. Because the tails are hydrophobic, they basically stack together to exclude water, while the phospholipid head groups, which are hydrophilic, line the outer part of the cell, the outer layer of the cell, and also the inner part of the cell, producing a bilayer of lipid which excludes water. The membrane proteins perform most of the membrane-specific tasks, and these can be receptors, transporters, so on and so forth.